Once the food products of interest have been identified, analysis can begin. To extract the DNA, take 2 clean screw cap tubes, and into each of these, transfer 500 microliters of purchased DNA isolation reagent, making sure to pipette the mix up and down between aliquots to evenly mix the reagent. Label one of the tubes non-GMO and the other test. Next, weigh out 0.5 grams of certified non-GMO food and place into a clean mortar. Add 2.5 milliliters of distilled water and grind with the pestle for 2 minutes to form a slurry. Add another 2.5 milliliters distilled water and continue grinding the slurry until it becomes smooth enough to pipette. Pipette 50 microliters of the known non-GMO slurry to the non-GMO labeled screw cap tube containing the DNA isolation reagent. Now add 50 microliters of the test food sample slurry to the screw cap tube marked test. Vortex the 2 tubes containing the food samples and DNA reagent for 1 minute. Next, place the tubes in a water bath at 95 degrees for 5 minutes. Finally, place the tubes in a centrifuge for 5 minutes. Upon examination, a solid pellet should be formed at the bottom of the tube. If a pellet is not formed after 5 minutes, centrifuge again for 2-minute intervals until a pellet forms. Samples can now be used immediately for PCR or stored in a refrigerator for up to 1 week. First, number 6 PCR tubes. These numbers will correspond to the tube contents listed in the table. Place each of the labeled PCR tubes in a microtube holder with caps open. Using a fresh tip for each addition, add 20 microliters primer indicated in the table to each PCR tube. Next, add 20 microliters of the DNA samples indicated in the table to each PCR tube. Pipette up and down to mix. For pelleted samples, be sure to pipette only from the supernatant and avoid the solid pellet at the bottom of the tubes. Finally, place the PCR tubes into a thermal cycler and program it to cycle through the heating and cooling steps indicated in the table. Place a 3% agarose gel into the electrophoresis chamber with the wells closest to the cathode end. Add TAE buffer into the chamber to cover 2 millimeters above the gel tray. Collect the PCR tubes from the thermocycler and place them in a microtube holder. Using a fresh pipette tip each time, add 10 microliters of purchased DNA loading dye to each sample and mix well. Using a fresh tip each time, load the wells of the agarose gel with 20 microliters of molecular weight ruler into one well and 20 microliters of each sample into subsequent wells, as indicated in this table. Set the electrophoresis chamber to run at 100 volts for 30 minutes. Once the gel has finished running, carefully unplug the gel chamber, remove the tray, and slide the gel into a staining tray. Immerse the gel in purchased 100x gel stain for 5 minutes, shaking the tray gently to distribute the stain. Once stained, transfer the gel into a washing container and rinse with tap water for approximately 10 seconds. Destain by washing 3 times in warm tap water for 3 minutes each, with gentle shaking for best results. If necessary, continue destaining in warm water until the desired contrast is reached. The presence or absence of a 200 base pair band in lane 4 indicates whether the test food contains GMOs. The plant primers determine whether plant DNA was successfully extracted from the sample. The non-GMO food control is an indicator of false positive results, should they occur. If the non-GMO food control comes out as positive, it means the PCR was contaminated at some point. The GMO positive template control is an indicator of false negatives. If the GMO-positive template control does not amplify, there is a problem with the PCR reaction, and a GMO-negative result from the test food can't be trusted. Gels can be placed on a white or yellow paper to provide contrasting background to highlight DNA bands, or increased contrast can be achieved using a UV light box. The ability to test foodstuffs or products for genetically modified ingredients is pertinent to a number of scientific or regulatory applications. There is some evidence that transgenic DNA from genetically modified crops can become introgressed into the genomes of wild crop populations. For example, pollinators may facilitate this transfer by fertilizing wild-type plants with pollen from a genetically modified source. This is a concern, as the impacts of the spread of these inserted genes on ecosystems as a whole are not well understood. PCR testing of wild populations can provide data on the potential spread or successful containment of genetic inserts. Lack of labeling or incorrect labeling of genetically modified ingredients can be a consumer concern. This may be due to the consumer's preference of consumption or potential introduction of allergens during the GM process, though the latter is controversial. In some countries, genetically modified ingredients are required to be listed on food packaging. Regulatory boards in such countries may use PCR testing to check the accuracy of food labeling. Where the genetic modification introduced to a crop confers pesticide resistance, some studies have raised concerns about the production of superweeds. Essentially, these can be any plant not initially targeted for modification that obtains the resistance to pesticide through mechanisms such as introgression or hybridization. These plants may then be able to spread more successfully and be harder to control than those without the insert. PCR testing for genetic modification can help to highlight and track such potential populations to determine if this spread could prove problematic. You've just watched Jove's introduction to testing for GMO foods. You should now understand the principles behind identifying genetically modified foods using PCR, how to extract and amplify DNA from food, and how to determine if your food sample has been genetically modified. Thanks for watching.